Chapter 7 is on Mendelian and quantitative genetics, and this is a very interesting chapter. I think a lot of you will find this neat to kind of see how your genes and your characteristics have been inherited from your parents. The science of genetics explains the stability of inheritance, so why you are human, as are your parents, and also the variations between offspring from one generation to the next. So why you look different from your parents or why you look different from your siblings. Until the late 1800s, most plant and animal breeders believed that traits were inherited by what they called a blending concept of inheritance. The blending concept of inheritance states that an offspring's genetic makeup is intermediate to that of its parents. So basically, like if you crossed a white flower with a red flower, you would end up with a pink flower. You'd end up with an intermediate between the two parents. But this theory did not really explain observed inheritance pattern. So why would a baby with one parent who has brown eyes and one parent who has blue eyes not always have hazel eyes, not always have an intermediate between the two parents? The breeders mistakenly attributed these variations to instability of genetic material, and this was obviously incorrect. Our genetic material is actually quite stable, but that was what they blamed. What they observed in nature was on instability of genetic material. Gregor Mendel was an Austrian monk who lived in the mid to late 1800s, and he developed the particulate theory of inheritance after performing a series of very creative experiments in the 1860s. Mendel's theory of inheritance is called the particulate theory because it's based on the existence of minute particles of hereditary units that we call genes. And what he believed was that inheritance involved the reshuffling of the same genes from one generation to the next. He worked with a garden pea as his experimental material, and he chose this plant for a lot of different reasons, but mainly because it had a short generation cycle, so he could observe changes very quickly from one generation to the next. He could also pollinate it hand, so he could control what the offspring looked like and what the crosses were between the parents. He observed characteristics such as stem length, pod shape, seed color, seed shape, flower position, flower color, pod color, things like that. And when he crossed plants, when he crossed parental plants, he observed either dominant or recessive characteristics, but no intermediate ones. And so the results of his experiments led him to certain conclusions that became the laws of inheritance. Mendel's particulate theory of inheritance is made up of the laws of segregation and the law of independent assortment. These laws describe the behavior of the units of heredity as they are passed on from one generation to the next. He did not know about chromosomes and genes, so he didn't really name the units. He just called them units of heredity. And most of our modern genetics today is actually based on his theories, which have withstood the test of time and been supported by a lot of different experiments from different scientists. The laws of segregation state that each individual has two factors for each trait. The factors will segregate or separate during the formation of gametes. Each gamete contains only one factor from each pair of factors, and then fertilization gives each new individual two factors for each trait. We know now that all of these things are basically true. You have a pair of chromosomes that carries alternate versions of a specific gene. These alternate versions are called alleles, and those chromosomes will separate during meiosis and go into different gametes, so that each gamete has only one allele for a trait. When two gametes come together, the new fertilized zygote has two alleles for each characteristic, one from each parental gamete. But what's amazing is that Mendel figured all this out just based on his observations of pea plants. He did not know about chromosomes, genes, and alleles, and so that's what's truly astounding is how he was able to come to these conclusions just based off of what he observed. His law of independent assortment states that each pair of factors will segregate or assort independently of the other pairs. So all possible combinations of factors can occur in the gametes. We know this is true as well because during meiosis, the way the chromosomes align during metaphase 1 determines which chromosome and therefore which alleles go into which gamete, and they do align independently of one another on that metaphase plate. The law of independent assortment applies only to alleles that are on different chromosomes. Clearly, alleles on the same chromosome would stay together unless they're mixed up by the process of crossing over that was discussed previously. Keep in mind that each chromosome does carry a very large number of alleles, so some traits we observe do seem to be inherited together. Let's go through some basics genetics now. We've already covered this some, but remember that chromosomes hold your genetic information in the form of genes, which are specific strips of DNA that code for certain proteins that manifest as characteristics such as hair color, eye color, and height, for some examples. Alternate versions of a gene are called alleles. A dominant allele is able to mask the expression of the other allele, which is called the recessive allele. The dominant allele is typically identified by a capital letter, and then the recessive allele is identified by the same letter, but it's the lowercase letter. So, for example, if we're talking about a pea plant, you'll see this, this shorthand. The allele for tallness is a capital T, and then the allele for shortness is a lowercase t. So tallness is dominant to shortness in a pea plant. When an individual or an organism has two identical alleles, it is considered to be homozygous, homo meaning same, so same alleles, homozygous. If both the alleles are dominant, the organism is homozygous dominant. If both the alleles are recessive, the organism is homozygous recessive. So homozygous dominant would have two uppercase letters, and homozygous recessive would have two lowercase letters. When an organism has two different alleles for a gene, then it's called heterozygous, hetero meaning different. So the allele that's expressed in that combination is going to be the dominant allele, because remember, the dominant allele by definition can mask the recessive allele. So a heterozygous individual would have, the shorthand version there would be an uppercase T and a lowercase t. It's a combination of two different alleles. The genotype of the individual is the genetic makeup of that individual. So it's all the alleles that that person receives at fertilization. For example, the genotype for a tall pea plant, remember tallness is dominant to shortness, could be either homozygous dominant, so two uppercase T's, or heterozygous, an uppercase T and a lowercase t. The phenotype of the individual is the physical manifestation or the outward appearance of that individual. So the phenotype of an individual that has a genotype that is homozygous dominant or heterozygous, so uppercase T, uppercase T, uppercase T, lowercase t, would be a tall plant. The phenotype of a homozygous recessive individual would be a short plant, so two lowercase t's would be a short plant. We've been talking about so far are autosomal patterns of inheritance. So that is patterns that are not involving the sex chromosomes. Remember, your autosomes are all of the chromosomes other than your sex chromosomes. In autosomal patterns of inheritance, if you have a dominant genetic disorder, the normal allele is lowercase, so the normal allele is recessive, and then the abnormal allele is uppercase or dominant. So any individual that has at least one dominant allele, so that would be the homozygous dominant and also the heterozygous individuals, would then have that disease. There isn't a silent carrier state. If you have the allele, you manifest with the disease. There's nobody that's carrying the allele that doesn't have the disease. There's no silent carrier state. In autosomal recessive disorders, the normal allele is uppercase and the abnormal allele is lowercase or recessive. So only individuals with both recessive alleles, that would be the homozygous recessive individuals, would then have the disorder. You can have a carrier state because heterozygous individuals would not manifest with the disease, but they could potentially pass it on to their offspring. If you know the genotype of the parents, you can use a Punnett square to predict the genotype and the phenotype of the offspring and the chances of an offspring inheriting a specific characteristic or a specific disease. So in a Punnett square, you put the alleles of one parent on one side of the square, so like on the top of the square, and then the alleles of the other parent on the side of the square. And then inside the square, you write down the possible combinations of alleles in the offspring based off the parents. In this example above, one parent has a homozygous recessive genotype, so that would be two lowercase t's, and you can see that written up above the top of the square. and then the other has a heterozygous genotype, and that's written to the left-hand side of the square. There's one uppercase T and one lowercase t. When you do the square, you combine those alleles. You can kind of see the arrows indicating where you would write in the offspring combinations, but the chances of the offspring with this cross would be 50% heterozygous and 50% homozygous recessive. So half of the offspring have that heterozygous combination with the uppercase T and the lowercase t, and then half have the homozygous recessive genotype with two lowercase t's. So that would mean that half would be inheriting a disease. If we're talking about a dominant genetic disorder, then half of them could potentially inherit it. An example of that is Huntington's disease, which you will come across as you read in your textbook. And so if we were talking about Huntington's disease with this Punnett square, you would have one parent that has the disease, and that would be the heterozygous individual, and then one parent that does not, which is the homozygous recessive individual. And in that situation, every single offspring has a 50% chance of inheriting the disorder from the parent. And this does raise an interesting point, which is, although Punnett squares can give you the probability of what would happen in offspring, chance has no memory. So in the previous example, every single child would have a 50% chance of inheriting the disease. So you could end up with every child having the disease, or you could end up with no child having the disease. Just because the firstborn child does not have the disease doesn't mean that the second-born child has a greater or lesser chance of inheriting it. Every single child has the same chance, a 50% chance of inheriting the disease. The last type of inheritance we're going to talk about in this chapter is called polygenic inheritance. In polygenic inheritance, a trait is controlled by more than one set of alleles. The alleles can be found on different chromosomes, and each dominant allele can have an additive effect on the phenotype, or make the phenotype more obvious or stronger. Many investigators are trying to determine what percentage of various traits are due to nature or inheritance versus nurture, or what percentage is due to the environment. Identical twins that have been separated at birth are more similar in their intellectual talents, their personality traits, and even their levels of lifelong happiness than are fraternal twins that are separated at birth. But as we've discussed before, sample size can have a big effect on the results of a study, and there just simply aren't that many identical twins that have been separated at birth for scientists to study. Your textbook has a really good discussion on the nature versus nurture debate that you should definitely read and understand at the end of this chapter.